Antonio, for this very kind invitation. It's a pleasure for me to be here to share with you some works we have done in my group in Pisa on the modeling of protein-light interaction. So I share my screen.
Yes.
Okay. Can you see?
It's coming. Yes, it's here now.
Okay.
Yes.
So, well, I think that I don't have to convince you that light is very important for biological function, and all organisms can sense light and use it for different scopes. Photosynthesis is a very famous example, but also photoreception, signal transduction. And the point is that this interaction with light goes through pigment protein complexes. I call them pigment protein complexes in the to say that, to underline that they are not just protein, but they are proteins containing a chromophoric unit, which can be a single molecule, a single chromophore, or an aggregate of chromophores. So these are composite systems. And the problem of modeling the effect of light on these proteins is the reason of is complicated because it is a multiscale problem in space and time. So in space is clear because you have the different scales. You have the chromophoric unit, the scale of the chromophoric unit. You have the protein, and then you have the environment around. But then there is also there are the scales in time, because you have the fast process, the absorption of light, and the photochemical or photophysical processes that happens in the chromophoric unit, which is fast. But then there is a cascade of events that covers different time scale, going from nanosecond, microsecond, millisecond, and so on. And in these scales, different scales, there can be different structural changes inside the protein, until even some conformational changes in the protein. So the idea is that one can use quantum mechanical techniques combined with classical techniques to cover these many scales in time and space. So in terms of the multiscale problem in space, a very effective approach used by many people is to combine quantum mechanics with molecular mechanics. Another approach is to use continuum solvation models that I work at the continuum solvation model a lot, but continuum solvation models are not accurate enough to describe a heterogeneous system like proteins. So we prefer to use an atomistic description, even if classical, but still atomistic. And so molecular mechanics is the best choice. So usually what people do is to combine quantum mechanics to molecular mechanics using the so-called electrostatic embedding. So the QM system feels the classical system like point charges, which represent the classical atoms. But there is something more. This something more is a mutual polarization effect. So if you want to include mutual polarization effect between the classical and the quantum part, you have to allow the force field to be polarizable. So there are different formulations of polarizable embedding. The one that we have developed in our group is based on the induced dipole. So the idea is that each atom is described by a fixed charge or a fixed multipole plus a polarizability. So in the presence of electric field, this polarizability generates induced atomic dipoles, and these atomic dipoles can interact, can respond to the quantum mechanical system. And the quantum mechanical system can feel the effect of the classical system. So in this way, we can introduce both electrostatic and mutopolarization effect, still keeping an atomistic level. Why we need the polarizable embedding? So in our case, to have a polarizable embedding is important because we are interested in photo-induced processes. So when you excite a chromophore inside a protein, you go from ground to an excited state. So we expect that the environment will polarize differently when in the ground, when the chromophore is in the ground, and when the chromophore is in the excited. If you don't have a polarizable embedding, you cannot take into account this effect. But there is more. When you do an excitation, there is another dynamic response of a polarizable embedding that depends on the transition. So depends on the transition density. And also this effect is important. And if you don't have a responsive environment, you cannot take into account this. So just to give you an example of this kind of response. A simple example. So we took retinal, which is the chromophore present in rhodopsin, and we did a simple calculation. So we first of all did QMMM molecular dynamics. Another group did the QMMM molecular dynamic trajectory. We took different configuration along this dynamic, and we computed excitation, vertical excitation, in different models. So the first model is the isolated chromophore retinal, so without protein, but with geometry calculated during the dynamics. So you can have different geometry of the retinal and different excitation energy. Then we switch on the effect of the protein with an electrostatic embedding, and what we see is a blue shift, a significant blue shift. Then we switch on the polarization of the environment, but just in the ground state, and we don't see a change with respect to the electrostatic embedding only formulation. But then when we switch on the effect of polarization also in the excitation process, we see that we have, we go back to the red. So the final excitation energy are very different from what you obtain in an electrostatic embedding. Can we explain this? To explain this, we have compared a full QM model with a QM MM-Pol model. So we start from the isolated retinal, this is blue, and then we add, first of all, a counter ion, the close by counter ion, which is very important in the response of the retinal. And we compute this system quantum mechanically, this is the blue excitation energy, or in a QM MM-Pol version. So if you just add this, you see a blue shift, exactly what was seen in the MM formulation. Why? Because the counter ion stabilizes the ground state a lot, and so you have this blue shift. Then when you enlarge the QM cluster, so to include the other residues, however, from this value, you go back to the red, and this is reproduced in our QM-MM-Pol formulation. Of course, if we introduce, when we introduce all the residues, we can do this description only at MM-Pol level, because at the quantum mechanical level is too much. But in this way, we have seen that the red shift is induced by a lot of a combined effect of all the residues around the chromophore. And what is important is that our polarizable MM will reproduce a full QM picture. So let's go a step further and let's focus on the problem in time. So now we have this problem of combining different time scales. Of course, if you want to sample the slow scale, so the fluctuation in the protein structure, you have to go in the microsecond or more time scale. So you necessarily have to use classical MM molecular dynamics. But if you want to study a photoinduced process, you have to recover a quantum mechanical description. So one strategy that I call sequential strategy, what does? It uses the classical MD configurations, as I did before, and compute QMMM or QMM-Paul static calculation on a larger set of different MM configuration. In this way, we can take into account the sampling of the protein, but we lack the coupling between nuclear and electronic motion, because the dynamics is fully classical, and then we compute static QMMM calculation. So another alternative is an integrated strategy. So we still use a lot of MM configuration, and starting from this configuration, we switch on QMMM or QMM-Paul dynamics. In this way, we recover the coupling between nuclear and electronic motion, because now the chromophore is treated, the dynamics of the chromophore is treated at QM level. This integrated strategy, we have implemented an interface between Tinker, which is the driver for molecular dynamics, and Gaussian, which is the software for QM-Paul calculation. So we have implemented the analytical gradients, the forces, and we have implemented all this stuff in a linear scaling framework, so that it can be applied to very large system. So let's see an example. As an application of this, I present a study we did a few years ago on the color change in lobster shell. So when you cook a lobster, the lobster shell goes from blue-black to orange-red. The reason for that is that the color is due to a pigment, which is a carotenoid, astaxanthin, and when it is in the protein, the excitation energy is correspond to blue-black. But when you cook, the crustaceanine protein is denaturated and astaxanthin is released. So now it's in an unbound form, and its absorption is shifted to orange. In the literature, there are many explanations of this shift, large shift, being suggested, but none of them are really robust. So what we did is to try to apply different models to try to understand. So this is the experiment. We have the unbound is our reference, and in the protein, there is a shift of 0.5 eV. The first guess, we used a non-polarizable MM description using the crystal structure of the crustaceanine. In the crystal structure, the protein is dimeric, and so you have two protein with two astaxanthin, and what we see at MM level is are two excitation energy very different, one from the other. If we add polarization, still in this crystal structure, we have a shift with respect to electrostatic embedding, but still we have a very large difference between the two monomers. Why? And this is not what we observe experimentally. So what's wrong in this model? So in order to understand, we said, well, maybe the problem is this crystal structure, and we need to go beyond that. So we need to introduce dynamics. We need to take into account the fluctuation in the protein structure. And as I said before, one strategy to do that is to do a full dynamics and to take different configuration. But here the problem is the chromophore. So in this case, the chromophore is a carotenoid, so a very long conjugated system. And we know that normal molecular mechanic, conventional molecular mechanic force field are not accurate enough to correctly describe such a large conjugation. And what can happen if you have a wrong force field is that your system sample a potential energy surface which is wrong. So when you do excitation energy on QMMM excitation energy on structure obtained with the wrong force field, you get wrong excitation energy. So in order to go beyond this, we need the integrated approach I presented a few slides ago. So what we did is a QM pole dynamics by using the interface between Tinker and Gaussian of the Kruster-Chanin dimeric protein. When we do that, we see that during this dynamics, the two astaxanthin in the two monomers explore different local field, and they give a range of excitation energies that, when considered all together, give an average, an effective excitation energy that, when compared with experiment, will explain the observer shift. So our explanation of the change of color in lobster is that the astaxanthin, the carotenoid in the protein, feel the effect of the protein both in terms of electrostatic fields, but also in terms of effect on the geometries that you can correctly reproduce only using a QMMM dynamics. So far so good, but what about more complex scenarios? As I said at the beginning of the presentation, a very important interaction of light with pigment protein complexes is photosynthesis. In particular, the first part of photosynthesis, the so-called light harvesting part of photosynthesis. So in light harvesting, what happens? You have a photosystem, which is a super complex made of different pigment protein complexes, the biantenna, which are pigment protein complexes which absorb light and transfer the absorbed energy to another pigment protein complex, which is the reaction center, where this energy is used to activate all the chemical reaction that produce photosynthesis. So the absorbed energy by the biantenna is rapidly transferred to the reaction center. And to have this transfer, which involve long distances, so tens of angstrom, we need an energy ladder. So we need an energy ladder in order to transfer the energy without dissipation. How in nature this energy ladder is created? In a very cheap way. So what nature developed is the idea of an excitonic system, so a multi-chromophoric system. So by using one type of chromophore, but putting many chromophores together, it creates new excited state called excitons, which are a different energy with respect to the excitation of the single chromophore. So using one single chromophore, but putting many chromophores together, you create a set of excitation at different energy. So you create an energy ladder. As an example of this strategy, in our group, we have studied a lot bacteria. So when people think about photosynthesis, usually think about plants. But actually, photosynthesis started with bacteria that then, through evolution, went to plants. So it's very interesting to study photosynthesis in bacteria. For example, in purple bacteria, we know a lot of these bacteria. we know the supercomplex, which contains the reaction center and in green the biantenna, which are called light harvesting 2 complexes. And these antenna are very unique. So they are very symmetric. So they are made of 2 aggregate of bacteriochlorophyll, 2 rings, one made of 18 bacteriochlorophyll and one made of 9 bacteriochlorophyll. And these bacteriochlorophyll are embedded in a symmetric protein matrix. So in order to describe this very large chromophoric system, you cannot just apply TDDFT of big system. So you need to introduce an approximation. So you reconstruct the excitation of the full multichromophoric system by an excitonic model. The idea is that you can reconstruct the full system calculating the property of each bacteriochlorophyll and the coupling between bacteriochlorophyll. The coupling is the interaction between excitation localized on different bacteriochlorophyll. So if you do this approach, you can simulate a very large system because the cost is limited. What is very important, however, in order to have a correct picture, is to introduce the effect of the environment both on the excitation of each bacteriochlorophyll, which is called site energy, and the coupling between excitation. In particular, in the coupling between excitation, that is generally described as a Coulomb interaction between transition densities localized on each bacteriochlorophyll. If you neglect the polarization effect of the environment, you overestimate this coupling because the environment, in a sense, screens through polarization the Coulomb interaction between the bacteriochlorophyll. And with our polarizable MM, we can take into account this. So if we apply this excitonic model, we can reconstruct all the excitons. And we see that because of the geometry of the system, there are 2 groups of exciton. One group of exciton is localized on the one ring called the B800 ring, made of 9 bacteriochlorophyll, and the other set of excitons is delocalized on the other ring, the so-called B850 ring. And because of the symmetry of the system, only few excitons are bright. So when you do absorption of this multichromophoric system, you don't see many excitation, but you see only 2 excitation, 2 peaks. One corresponding to the bright state localized on the green ring, let's call it, and one corresponding to the bright state localized on the B850. The name of this ring is due to the wavelength characteristic of the 2 excitation. So if you look at the experimental data, you see that the absorption spectra of this system is temperature dependent. In particular, going from very low temperature to room temperature, you see a shift in the B800 in the low energy excitons, so the B850, while the other peak is insensitive to temperature. But there is more. If you measure the time needed to transfer energy from the high energy exciton to the low energy exciton, you see that this transfer becomes faster when you increase the temperature. So a 60% more rapid transfer, faster transfer. Can we explain this behavior? So first of all, we need to include temperature. To include temperature, we do dynamics, because we have to include the fluctuation of the protein. I recall that these proteins are membrane proteins. So you have to do the simulation in a model for the membrane and applied excitonic calculation along these dynamics. In this case, we use classical, the system is too large, so we use a classic dynamics, and then we do QM pole excitonic. If you do that, you discover an interesting thing. So if you start from the crystal structure, which is very symmetric, you see that, so no disorder because of the symmetry, you see that the exciton in the B850 ring is extremely delocalized. So all the bacteriochlorophyll are involved in the exciton. So the exciton length is maximum. You can compute the exciton length through the expansion of the exciton on the basis of the bacteriochlorophyll. So when you have 18, means that you have all the bacteriochlorophyll involved in the exciton. When you do molecular dynamics, you take into account fluctuation and disorder, you get a distribution of the exciton length, a distribution that has a maximum around 8. So it means that in the average, the exciton length, the exciton involve 8 bacteriochlorophyll, not 18. So this means that thermal fluctuations induce localization in the ring. And this localization is the reason why we see this shift in the spectra. Because if you localize, you get a smaller bandwidth of the set of exciton, and you have a blue shift in the peak of the signal. So we can explain the spectroscopy. But what about the time, the transfer time? So to calculate energy transfer time, we need a model. So I don't enter into the details, but you can reconstruct transfer time using an approach which is a formalism which is used in open quantum systems. So you assume that your excitonic system is coupled to a buff of quantum oscillators. You solve the quantum master equation in a perturbative way, and you get the transfer times. There are different perturbative approach, the so-called Foster approach, the so-called Redfield approach. In our case, we used a modified Redfield approach, which is suited for this kind of system. So by doing that, what we get? So this is an experiment. So in the experiment, the transfer time from the one ring to the other is of the order of 1.2 picosecond at very low temperature to with that when you increase the temperature, you go to room temperature, the transfer time is shorter. So in our calculation, indeed, we see this shortening of the transfer time that, again, we can explain in terms of the localization that we have seen in the spectroscopy. So this result is very interesting from a biological point of view, because it tells us that the ring structure of this system is the secret, because of this ring structure makes the transfer process robust with respect to thermal fluctuation and disorder, which is the typical regime of a biological system at room temperature. But this system is very interesting also for a different point of view. So if you these bacteria can adapt very well to different light conditions. So when the bacteria is grown under light condition, high light condition, the ratio between antenna and reaction center is high, because you have to take you have a lot of energy that can be absorbed. But when the system is grown under low light condition, there is a change in the ratio, because you have to maximize the light harvesting, because you have fewer photons available. So the photosystem changes in terms of composition. But there is more. Not only the photosystem change in terms of composition, but the single antenna change. So the single LH2 antenna, if the bacteria is grown under high light condition or low light condition, is different, and the spectroscopy of the antenna changes. So previously we saw this highlight, the absorption of the highlight form, and you see that there is a large overlap between the absorption of the antenna with the absorption of the reaction center that is surrounded by another antenna. While when you grow the bacteria in a low light condition, the absorption of the antenna is changed. You see, there is a very important shift, and this shift in the absorption, we think that is due to the fact that in low light condition, you have to avoid any possible back transfer from the reaction center to the light harvesting, because in this way you lose energy. So how is it possible, this shift in the response of the light harvesting system? It's very intriguing, the problem, because if you look at the crystal structure of high light and low light forms, you have exactly the same multichromophoric system. So the multichromophoric aggregate is exactly the same, but what changed? So in order to understand what is changed, we have to see how this antenna is made. So the antenna has a modular structure, so is obtained by a replication 9 times of a constitutive module, which is made of 2 protein chains binding 3 bacteriochlorophyll. If you replicate circularly 9 times, you get the 2 rings that I presented before. So the bacteriochlorophyll are anchored to the protein chains through histidine magnesium interaction in the B850 ring, through van der Waals content in the other. However, there is more. There are 2 hydrogen bonding interaction between the bacteriochlorophyll and the residues around. This is the high light form. What happened in the low light form? So in the low light form, there is a mutation of the residues around the bacteriochlorophyll, and this implies a change in the hydrogen bond interaction between the protein and the multichromophoric aggregate, the bacteriochlorophyll. In particular, you lose nitrogen bonding in one of the bacteriochlorophyll of the trimer that is replicated. So if you lose a nitrogen bonding, you expect that there is a shift in the excitation, because there is no more this hydrogen bonding. So the excitation energy on this bacteriochlorophyll in the low light or in this bacteriochlorophyll in the high light will be different. So the excitonic picture that you get will be different. In fact, if we compute the excitons of this, imagine the full ring, we see a shift, a blue shift, so qualitatively correct blue shift passing from high light to low light, but this shift is too small. So what is missing in our model? Again, what is missing is the dynamic effect. So again, you have to repeat the dynamic picture of the high light and the low light, and if you do that, you realize that during this dynamic, the effect of this dynamic, of the disorder I talked before, is different in the 2 forms because of the different hydrogen bonding interaction. In particular, what we see is that in the low light, there is a larger orientational disorder that with respect to highlight. And this larger orientational disorder is reflected in the couplings. So what happens in the low light is that the inter-unit couplings are reduced with respect to the highlight form. So if you take into account this dynamic effect and recompute the excitons, what you get is the following. So on the left, there are the experiment. Here is in centimeters -1 before it was in nanometers. So in red, you have the highlight form. In blue, you have the low light form with the blue shift. And on the right, you have the simulation, including both the static and dynamic effect I have there just presented. So the conclusion from a biological point of view. So the chip strategy used by the protein to control the exciton is not to change the multichromophoric system, but just to change a few residues which interacts through hydrogen bonding with the bacteriochlorophyll. And so through this hydrogen bonding effect, the protein controls the position and the nature of the excitons. So it controls the energy transfer process. So to conclude, what come out from all these analyses is that light-induced processes in proteins are the results of a delicate interplay of chromophore protein interaction and coupled dynamics. So the protein acts through electrostatic field, polarization effect, and chemical and physical constraint like hydrogen bonding, binding pocket constraint, and so on. So what we can do to have a even more complete picture? So what we have to do is to move from protein-induced processes which are based on chromophore with well-separated electronic states to more complex picture which require non-adiabatic dynamics. So in our group, we are now working in the implementation of a non-adiabatic dynamics coupled to polarizable embedding. And the other very important point, I haven't had time to present this, is to study other light-induced processes which involve a very large conformational change in the proteins. And when you have a very large conformational change in the proteins, you have to go beyond the conventional MD, you have to introduce enhanced sampling techniques. And so another direction of development in our group is to combine enhanced sampling technique with the QM and Paul description. So to conclude, I want to thank all the people in the group, in particular Filippo, who is the responsible for the development of QM and Paul techniques, together with the group of Jean-Philippe Kemal in Paris, and all the people, Lorenzo, who worked a lot on this light harvesting system, and all the people in the group. And I want to thank you for your attention.
Thank you very much. Very nice talk. Thank you very much.
Thank you.
Thank you. It's impressive the amount of work you can do and the quality of your work. Thank you very much. So now you can ask, please. Just raise your hand. I don't know if you can see everybody, but I think, or just place your question. Okay. Questions, please. Carlos, yes, Carlos, please.
Yeah, hi everyone.
Hi.
Hi, Benedetta. Very nice to see your face and to hear you giving a talk for the first time from Cleveland. I have read many of your papers. I have used many of your development, and it's a great pleasure to see you. Excellent talk.
Thank you.
It's always good to put a face on
It's
in 2D, but it's moving. I have seen you, of course, in pictures, but at least I can see you now moving into dimensions. I look forward to opportunity to see you in 3D. I was wondering, so clearly in your talk, hydrogen bonding play a very important role in controlling this the coupling, excitonic coupling, and also the energy transfer. In the polarization method that you develop, can you also include specific hydrogen bonding interaction with, or how you include that? Can you do it at the MD level, or so?
In these results I presented, the hydrogen bond interaction are treated at the QM and pol level. So the bacteriochlorophyll are QM, and the hydrogen bonding residues are polarizable MM. So so in our case, the effect of the hydrogen bonding is treated at the hybrid level. We haven't we haven't done some tests including the hydrogen bonding residues in the QM subsystem, but the picture is very similar.
Mm-hmm. Yeah, so I'm wondering if this polarization can be used to take in consideration specific hydrogen bonding interaction, for example, when you have solvation in like water instead of having a water molecules, that will.
The point is exactly this, in the sense that you can use this polarizable to have an atomistic description of the classical system. And so hydrogen bonding is treated really in this limit, okay? Mm-hmm. So for example, in continuum model, this effect is not there because you have a continuum classical system. Here you have still an classical but atomistic. And polarizable embedding are very good at describing hydrogen bonding effect.
Mm-hmm. Okay, yeah, that's good to know. Yeah. I appreciate that. If there was one of your slides I seen at the beginning, maybe slide 3, or where you were comparing gas phase and if you could please.
See, I come. Yeah, I have to share again.
Yes.
Here, you say. Is this one?
A previous one. Yeah, maybe the previous slide. Yeah, this one. Yeah. So it was very interesting to me to see that the vacuum at protein compared to the pol field basically just match the energies. Any explanation for that, or?
Yeah. The explanation is the one I gave you is here. So, okay, this is a single configuration, but you can do many configurations. So here is the retinal alone. So without any protein, okay? The excitation energy. Then if you add the residue which interact more, which is close, it's charged, so it's a counter ion, this one, you have a blue shift. But if you enlarge the protein, so you add more residues, the effect of the other residues, it was here, reduces in some sense, it goes in the opposite direction. So this is a blue shift because it stabilizes a lot the ground state, while the rest, adding the rest of the protein, do the opposite. It stabilizes the excited state. So you go down again. And so at the end, retinal in protein is very similar to retinal alone.
Yes.
Because I understood. It's a combination. is a combination of opposite effect.
I see. Okay. Yeah, that was because, yeah, I was wondering, well, is there is a need at the end to add all the environment if the gas phase in the vacuum calculation is predicting well. But I see is that
Yeah, but this is
just
a case you don't know at the beginning. You have
exactly,
check
yeah.
this.
So maybe for this particular
This
system, it
particular
works like
one,
that. Yes.
yes.
Okay. Yeah. Thank you. I appreciate that. Very nice talk.
Thank you, Carlos. Now, Paula.
Bene, thanks. Your seminar was wonderful, and all these beautiful figures also in these complex systems. And it's impossible to simplify them because they are very complex, and this complexity that gives these absorption effects.
Exactly.
So it's very hard to put everything together. These membrane proteins that you have to put the membrane to fix the protein.
The structure, yes.
The structure, because if without that, the protein may open everything. So
yeah,
it's very difficult. So I have a smaller question about the computational times with all these complex things, and about the selection of the functionals to describe the chromophores. Sometimes you use chemistry leap, sometimes omega, yeah. So they are range separated functionals. So they are selected because from I think sometimes for porphyrins they open too much the gap. So I don't know. And if they help with dispersion corrections also, or if you try to use something like that.
Okay, so the computational cost is difficult to say because you have different steps. In the sense you have to do a molecular dynamics long enough to have the sampling of the complex in the membrane. After that, you have to repeat the excitonic calculation for many configurations in order to have a correct picture, because you cannot use just one configuration because you miss all the effect of the disorder and so on. But what is good is this excitonic approach, because this excitonic approach is very, it scales very well with the dimension of the system, because what you have to do is a calculation on each bacteriochlorophyll with the rest treated classically at polarizable MM. And then combining this calculation to compute the coupling. So the cost is not so large. So the cost of each calculation is a TD-DFT calculation of on a single bacteriochlorophyll embedded in a polarizable MM. So it's not so costly. Then you have to reconstruct the coupling, but again, it's not so costly. So at the end, it's feasible. It's you can treat very large system, but because of this excitonic, because if I would like to do the full QM on the full multichromophoric system, no way. Okay, so you have to reconstruct in pieces. So concerning the DFT, this is an issue. So every time we have to check and so on. So bacteriochlorophyll, I haven't said, I was fast, but for the bacteriochlorophyll, we are interested in the so-called QI excitation. The QI excitation is the lowest in energy excitation. And this excitation is reasonably described by most of the functionals, I have to say. There are some differences, but not so large. However, when you go to carotenoids, for example, the carotenoid I presented in the lobster, there the situation is more complicated because there you need we see that a long-range corrected is better. And in particular, we saw that this omega B97XD functional works very well. It doesn't give you, it doesn't give you exactly the absolute value of the energy with respect to experiment. There is a shift, but it gives you correctly, in a correct way, the dependence of this excitation energy to the geometry, which is a fundamental issue. Because if you have a perfect functional that gives you the correct energy in the minimum, but is wrong in the other configuration, for us it's a mess. So we prefer a less accurate in terms of absolute energy, but a more robust functional. And CAM is less robust than omega in terms of this. But for all for any system, you have to check because it's different.
We made some calculations for cobalt porphyrin, and in fact, for Q bands, you can choose any functional, even that local. But
exactly.
when you have the metal, so everything comes strange. So it's very difficult to find that.
We are safe because we are interested in this QY excitation.
Yeah, nice. And if I may, another question about the emission. How can you treat that?
Yeah, we can do emission. I haven't presented the data, but you can. We can do geometry optimization of the excited state in QMM pole, but we can do more. We can do dynamics in the excited state. So we have implemented this interface between Tinker and Gaussian. So at the moment we can do Born-Oppenheimer molecular dynamics, but as I said, grounded excited. But as I said, we are now working on the non-adiabatic extension to do photochemistry, of course. But still, with the Born-Oppenheimer, you can simulate spectroscopies. And you can also simulate, I haven't had time to present, but you can also simulate excited state. For example, now we are studying an infrared spectra in the excited state. Nice. We can reconstruct from QM pole dynamics. So you can do emission, you can do circular dichroism, you can do,
Nice, thank you, thank you.
Thank you, Paula. There is another question by Giacomo Lodi. Is that right?
Yeah, correct. Hi, everyone. I would like to thanks Antonio for letting me attending to this seminar. It was very enlightening. And now I have a question, maybe one more for Benedetta. And I was wondering when you presented at the beginning of your presentation that you implemented the model, the introduction of polarizable embedding based on induced dipoles.
Yes.
I was wondering, how do you compute the induced dipole contribution? Is there a self-consistent solution of the problem?
Yes, surely it's a self-consistent solution. Yes. So in particular, it's a self-consistent when you do excited states. It's self-consistent also in the excited state.
Okay, thank you. And if I may ask another question, you said also that in computing the excitonic coupling, it is important to include the polarizable environment because of the screening.
Mostly, yes.
How much is the ratio between, if you take just the Coulombic part and the screening? Does it depend on the dielectric constant of the environment?
Yeah, it's a good point. So depends. There are two effects. I haven't. I was fast, of course. So the effect on the environment mostly screens, and if you use a simple model using a continuum description, you can demonstrate that this screening effect is around 1/2. There is this Foster model that use this 1/epsilon optical, which is around 2 to write. It's a classical model, but gives you an idea. So with our approach, we are more, let's say, complete in the description, because and so it's not 1/2 in this sense, because the effect on environment also is on the transition density. So if you don't use the screen, if you don't add the screening effect, but you introduce the effect of the environment on the calculation of the transition density, the coupling increases with respect to gas phase. So it's a combination of this increase due to the, you can imagine in a dipole-dipole way, no? In the dipole-dipole framework, the transition dipole increase due to the environment, so the direct coupling increases. But then there is this, the opposite effect of the screening, which reduces. So at the end, it's not 1/2, it's much less than 1/2, but it depends on the system. But in the limit is around 1/2.
Okay, okay. Can I have another one?
Yeah, another one.
Okay. You basically show throughout the presentation that you are studying excitonic excitation. Basically, they are localized, let's say, Frankel. But what happens if you have charge transfer events? Then is your model, let's say, the tight binding is appropriate?
Yeah, this is again, this is tricky, indeed. So in the calculation, in the results I presented, there are no charge transfer effect, so it's a flank pure. But we have implemented an extension, so to include the charge transfer effect, using the bytization technique, but we can include charge transfer effect. And we have seen that in particular in LH2, this system, for the ring where the chlorophyll are very close to each other, this charge transfer effect is not negligible. It shifts a bit the excitons. So this charge transfer effect in the excitonic system is a very open problem, because also in other light harvesting plants, for example, in other light harvesting system, there is this hypothesis that these charge transfer effect are into play not for light harvesting, but for photoprotection. So going back to the method, yes, you can include charge transfer effect. It's more costly, of course, because you have to do dimeric calculations, at least. But we can.
Okay.
It costs more.
Yeah, of course, of course. Okay. Thank you. I will stop here.
Okay. Thank you, Giacomo. Now, next one is Jonathan.
Hey, good afternoon, everyone. Good afternoon, Benedetta. It's a great pleasure to meet you. I, like another colleagues here, I also use some methods developed by you.
I hope that they work.
And thank you for the great presentation. It's very interesting. Photochemistry, photophysics is very interesting. I just have a curiosity. If it's possible to treat the KM part with multi-reference methods, for example, CASSF?
Yeah, in principle, yes. Yes, we have with Carles Curucce that Paula knows. We did an extension of our Amenpol formulation to CAS-CF. So it's possible. There is an issue because when you do a CAS-CF approach and when you solve for different state, the problem is that which polarization do you use for which state? Because each state is different. With the DDFT, this is not an issue. So there are different approximations that you can use, but yeah, you can extend it, and it's being extended to CAS-CF. Yes.
Cool. And I have just another question about the EET process. What is the methodology that you use to treat the energy transfer couplings?
Yeah, the methodology is based on this open quantum system approach. So we have the excitonic model, the excitonic system that I presented, and we think that this excitonic system, we assume, interact with a set of quantum oscillator. So you can reconstruct from this model the coupling, the spectral density is called, the coupling between vibration, the quantum oscillator are the vibration in some sense, no? So you can reconstruct the coupling between the excitation and the vibration, which are represented in terms of a quantum oscillator. And if you do that, you can there are some perturbative solution of this quantum mechanical problem that you can apply, and you obtain directly the transfer time. So what you need to calculate, in addition to the excited state, is this coupling between vibration, so your quantum oscillator, and excitation. So in this way, you take into account the quantum mechanically the effect of vibration on the relaxation of the excited state, no? And you reconstruct the transfer times.
Right. Oh, thank you so much.
Sorry, I cannot be too detailed, but it's not something that you explain in 2 words, because there are. But the idea is this: you have your QM-Pol excitonic model coupled to this quantum oscillator. Okay. And during the excitation, these 2 things are coupled, and they induce a relaxation of the excitons.
Okay, I get it. Thank you so much.
Thank you, Jonathan. Thank you. Now, Eric, please.
Benedetta, great talk.
Thank you.
I have a very specific question. Is it possible to use your method to study Dexter energy transfer in a very complex system where the energy transfer concurs with a structural change?
Dexter energy transfer is complicated because you need accurate QM methods, because it's based, it's not a Coulomb interaction, which is easy to describe. So there are exchange effect and so on. With this approach, no, it doesn't work, because it cannot be represented in terms of this transition density coupling. You have to, there are other techniques. We can use the same technique used for the charge transfer effect. So you do a dimer, and you diabatize, use diabatization techniques, and reconstruct the coupling. So it can be done, but it is less simple than this Coulomb term.
Thank you very much.
Yeah.
Is there any other question? No? So, I think, no. Yes, there is no other question. So, I just want to thank you again, Benedetta, because it was a very, very nice, and you, as you can see by the number of questions and different people asking you, congratulations for your work, and thank you very much for being with us. I hope to meet you again.
Yeah.
And we are going to keep this series of webinars in the second semester now. Okay. So you closed our semester.
Okay. Okay.
And so it's a with gold medal.
You go. There are vacation now, more or less.
Yes, in July we have 1 month for vacation.
Okay.
And the seminar is every 15 days. So we are going to close now, and we start again in August. So
it's a very good idea. This of
thank you. Yes, for us it's good because it's not easy to meet people from USA or Europe, and in this way it's very easy.
This is one of the few positive aspect of pandemic to have this web seminar, that it was not so common in the past.
No. We are just a bunch of bits, and so it's easy to teletransport from one place. So thank you very much, and I thank you everybody for being with us today. Thank you.
Thank you for the invitation.
Thank you. Have a nice weekend, everybody. Thank you.
To you.
Ciao
a tutti. Bye.
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 Bye, Carlos.
Ciao Paola.
Ti vediamo.
Tuo marito.
Grazie. Ciao. Ti vediamo. Saluti.
Ciao.
Ciao.